right, so thank you everyone for joining again. And before we begin today's call, I would like to go over just a few housekeeping rules, just the standard stuff. So I think I have the settings on here to mute everybody as they come into the room. But if you notice that your mic is not muted and there's a lot of background noise around you, please be courteous and mute your mics. I know we've had problems with that in the past, and we don't want to do any sort of inconveniences to our speakers, unless you have a question or something that is burning that you really you have to ask. I'm sure that we can accommodate that. As I said, I'm going to be kind of monitoring the call to make sure that anybody who is joining a little bit late can go ahead and get in. And if you have any problems with that, please either post on the Slack or contact me. I'll have my email up as well and going. But without further ado, I would like to introduce Sam Nicholls to the call. I think I'm going to just pass it over to him so that he can kind of give a little introduction of himself and start going with the talk about Climb COVID. So Great. Thank you, Sam.
Thank you. Thanks very much. I assume you can hear me and see my screen. I'm Sam Nicholls, and I'll be talking for about 40 minutes about the infrastructure that we've built here in the UK called Climb COVID, which is our sort of centralized metadata and sequence processing platform that's driven all of the national surveillance of SARS-CoV-2 here in the UK. So I'm Sam Nicholls. I'm a research fellow at the University of Birmingham, and just to clarify, this is not to be confused with any of the many Birminghams that you can find over the pond in the United States. I'm roughly smack bang in the middle of England, and at the University of Birmingham, I'm a research fellow, and really by trade, I'm a bioinformatician, which is probably a word that doesn't need to be defined in the context of this call. It's great to be talking about this work to so many bioinformaticians. And what I was doing while I was back in the office in the before times, I was a research fellow looking at metagenomics, and particularly we were looking at extracting interesting strains from stool. And we were doing that to see whether we could determine why, from a genomic perspective, fecal transplants in patients with ulcerative colitis could be successful or not. And that was looking really, really good and really promising, had some really exciting research until around March 2020. This graph probably needs no introduction. This is from the UK government website. On the x-axis is time, stretching what feels like a very, very long time now back into March 2020 up to the beginning of October this year. And on the y-axis is the number of positive SARS-CoV-2 cases in the UK by the date that the sample was taken. And we had our first wave back in sort of April last year, which felt terrible. And then we had another one at Christmas, and then January peaked. And now we're on track. Well, now we're in the midst of sort of a fourth wave. It's hard to really describe it as a wave because it's just been continuing since around June. Unfortunately, I just think we're on track for a really difficult winter. And this is a shame because I think it really speaks to our sort of level of pandemic preparedness, or rather our lack thereof, which shouldn't really be a surprise because as a species, humans have been surrounded by pathogens and epidemics for a very, very long time. And one of the sort of scientific advances that we've recently been able to harness to kind of take a look at how an epidemic is spreading is through genomic sequencing. And I don't think I need to spend very long talking about why it's really, really useful in the context of pathogen surveillance, but there's a really nice reviews article here that talks about how you can take a sample from a patient or group of patients from out in a community where a pathogen is spreading, sequence all the genomes of those pathogen samples, and then you can do comparative analysis of them to try and work out what the chains of transmission are and contextualize how the virus is moving around in the population. And the nice thing about this review paper is it talks about how that's been used in developing countries for Zika and yellow fever and HIV and all of those sorts of diseases, and how the stepping in with this genomics informed surveillance can actually invoke a positive public health response. So we wanted to do that here in the UK. So last year, we founded the Covid19 Genomics UK Consortium, which is lovingly known as COG UK. It's a national network of NHS organisations, all 4 of our UK public health agencies, so that's Scotland, Wales, England, and Northern Ireland. Around 20 or so different academic partners, the Wellcome Sanger Institute, and the lighthouse laboratories, which are high throughput mega laboratories that were set up by the government to do sort of mass high throughput diagnostic testing. And COG was initially supported by £20 million of funding from the UK Department of Health and Social Care and the UKRI, which is the sort of UK funding body. And COG kind of had three goals. Its job was to produce data by connecting a network of sequencing capable organizations to a network of sample collecting organizations. And then COG wanted to collect that data somehow by providing some unified system to transfer and store the genomes and any metadata about those genomes and samples. And then finally, we wanted to integrate that data into a single canonical data set by harmonizing the sequences and the metadata, and then making it available for analysis so that public health analysis could go and look at the data and actually, help drive decision making to curb the spread. There's not a huge amount to say on producing. You can imagine some sort of sampling network of hospitals, GP surgeries, vans that go around swabbing people on the street to see if they have COVID. And out of this sampling network on my diagram here on the left, these sort of funny pointy things are supposed to be sort of positive SARS-CoV-2 samples. And on the right-hand side, the little file-looking pictograms are representing the metadata about that sample. So where it was collected from, for example. And I think really what I want to highlight here, the sort of most powerful part of COG's distributed model is that when a sequencing organization joined the consortium, we did not demand that they do things in a particular way. That it's up to them how they want to do their extraction, their library prep, their sequencing, and their bioinformatics. So sequencing sites here are completely autonomous, and this really reduces the burden on participating. But not only that, it means that you're getting the sort of the expertise of that local site. Without having to burden them with following some new protocol that they might not get right or might not understand or whatever. So they can really hit the ground running and start delivering samples and metadata. And so for every sample, what we ask from them is a consensus sequence, so a consensus of the SARS-CoV-2 virus that was sampled, and then an alignment of the reads against the original sort of canonical Wuhan reference sequence. And then on the right-hand side of this diagram, some poor person at this sequencing site is responsible for managing and merging and munging the metadata into some kind of merged metadata format, which I'll talk about a bit later, but that's obviously a very important step as well. In terms of how samples got to COG, there were sort of two main routes that I've symbolized here. On the left, we have pillar one. These are sort of smaller hospitals and public health laboratories. And then on the right-hand side, we've got pillar two, which is sort of mass throughput community testing, typically through those lighthouse labs that I mentioned earlier. And they come into this sequencing pyramid. And the idea is at the top of the pyramid, we've got low throughput but fast turnaround sites. And at the bottom, we've got high throughput but slow turnaround. And the idea here is that we're getting kind of two sides of the same coin of the epidemic, of pandemic. At the top, we're getting small amounts of sequences turned around very, very quickly. So this means that we can actually deliver information on, is this an outbreak in this ward? Do we need to isolate everyone? Do we need to separate people? Do we need to, instigate something that's going to help break those small localized chains of transmission? And then at the bottom of the pyramid, these huge centralized sequencing facilities like the Sanger, they will be answering not so much the small scale stuff, but the big scale stuff, the large scale backbone of what does this pandemic look like at a national level. And they'll be taking thousands of samples and sequencing thousands of samples every single day. And it takes longer to do that, but it gives you through these sort of two means, we have the ability to investigate small outbreaks and like the large national picture. So once we've got that, we want to be able to collect this data. And this is sort of really one of the most interesting parts of the project for me, certainly from a bioinformatics perspective. It's really a sort of interoperability problem because we have all of these different sites generating data in slightly different ways. And we want to make sure that when we bring it all together, it's able to be used in a useful way. So that the data basically needs to have some sort of unified interface. But to really get this interoperability to work, we identified that we needed three things. So firstly, we needed a location that could be reached by everyone in the consortium that actually had the capacity to hold the sequences and the metadata for all of the samples sequenced by COG. And then secondly, that sequence and metadata needs to adhere as best as possible to some specification, and that specification needs to be applied evenly to every single sample submitted to the consortium. And then finally, we need to put it somewhere like a database so people can actually access and retrieve and update information quickly. And ideally, they should be able to get the data back out again. So in terms of location, we're incredibly well placed here in Birmingham. Birmingham is a founding member of the Cloud Infrastructure for Microbial Bioinformatics. It's quite possibly one of the largest compute platforms for bioinformatics. It's certainly the largest in the UK. It's used by about 85 different academic groups. And in Birmingham and Cardiff, who are two of the founding members, we've isolated sort of a piece of Climb. We've taken it out of the Climb platform and we've created something called Climb COVID, which has 0.5 petabyte of fast SSD storage, but we'll be upgrading that soon. All of it is hosted in our state-of-the-art warm water cooled compute data center. And then sort of the gist of how things work is we have a large shared login node that has 96 CPU and 3 terabytes of RAM to do basic analysis, querying the data set, having a look at the data. And then from that login node, people are also able to farm out jobs to the compute cluster. So there's plenty of storage for samples, plenty of room to do compute jobs and work, but we still need a home for the metadata. And before we can really talk about where the metadata goes, we need to talk about what it's going to look like. So I said that we needed a specification. And the reason for this to begin with is that the sequences are obviously going to have little utility without any corresponding metadata. If we can't contextualize the sample to a time or place, then really what good is the genome? So we decided very early on that our analysis pipelines will just not process sequences unless there's some basic metadata. But what should that metadata be? So we knew it was obvious from the beginning that we need to collect things like sample collection date and location, because that's really, really useful for building phylogenetic trees. But it was less obvious to us that some centers would actually not be able to tell us when a sample was collected. Even seemingly random identifiers for a sample could be considered identifiable by some organizations. So there was a whole problem about, oh, we can't share what this sample is called with you. We need to call it something else. In terms of collecting location, England has 6 metropolitan counties, 75 non-metropolitan counties, and London, the capital alone, is broken up into 32 different boroughs. So, if you want to say this sample is from a particular place, it's quite actually difficult to pinpoint where in England that actually is, because there's about 3 different ways of representing things at a county level. So you might think, okay, we'll collect postcode, outer postcode, which is like a part of a zip code, I guess. But this actually led to a legal obstacle course that took months to finish. And obviously, it's very, very important to take ethical things like collecting this sort of level of information seriously. But it was that caused a real bit of trouble for us, because it meant that we were not able to really distinguish where in quite large areas, in some cases, that a sample had come from. Ultimately, we had many, many meetings on what metadata to collect, but really we discovered that it was impossible for any field to be made mandatory. And I think my warning here, and many of you have probably encountered this, the true difficulty in collecting this metadata is really about what can be extracted given the local staff time and resources on the ground where these samples are being collected. There's no one-off solution. So because of that, we picked a very, very minimal specification. We asked for a sample name, so a unique sample name, a collection date, and if that's not available, a received date, so basically the earliest known date that this sample existed, and the country of collection, so that would be England, Wales, Scotland, or Northern Ireland, which is obviously a huge, huge geographical area. So ideally, we want something more like the county of collection. So we might say, okay, this is from Birmingham, but better yet, we want to know what the outer postcode, what piece of Birmingham is that from? And then other sort of contextualizing information, perhaps like patient age and sex. And then also sample CT values are quite useful for determining why some sequencing might have failed, from a quality control perspective. So we have this specification, but how can we collect it all together? We need some sort of mechanism for people to be able to upload data that's easily accessible from behind firewalls, shouldn't require any specialist applications to be installed, and it should be suitable for people, for non-technical people, because this was a huge, huge national scale effort with thousands of people involved, and not all of them had the computer savvy to be able to do this. We needed to make it as simple as possible to provide metadata. So dare I say it, at the intersection between intuitive and portable exists spreadsheets. They're easy to fill in, they're easy to share, and really almost everyone has access to some spreadsheet software. Every ancient machine in the dark corner of every public health laboratory has some horribly old version of Excel on it. And on the right here is just an example of what that might look like. We've got a column for each of the metadata fields and a row for each of the samples. So here there's a unique sample ID, a country, and a received date. But we've got these CSVs, but how can we collect, validate, and store the data within these spreadsheets? Obviously, we don't want to rely on embedded software-based validators because people might turn them off or ignore them, or they might have software that doesn't even support them. It's clearly not going to be feasible to collect and moderate these files manually. And even if it was, We don't want slow, clumsy humans getting involved in any part of this process, because we want the metadata to be, ingested into the data set immediately. And if there's problems with it, we want to know immediately. So this is really starting to sound like a job for computers. And this is kind of really where I come into the consortium. I built a piece of software called Majora. I think it's fair to say that Majora is the brain of Cog UK. It's a database that stores all of the Cog metadata about all of the samples and sequencing that's gone on. It's a website that shows limited pieces of that metadata and allows users to manage their accounts and things that the consortium can do for them automatically. And it's also a set of APIs that allow the database to be updated or queried. So it's effectively a national scale LIMS and then some. It was built for Cog from scratch using the Django framework. If you're not familiar, it's a Python-based rapid web development framework. Our first user was actually registered in just 3 days, and we unified the entire metadata process for every single site in Cog within 3 weeks. So I think by April 10th, this was up and running, April 10th, 2020. So there's 2 core classes of metadata held in Majora. We have artifacts and processes, and artifacts are things to be tracked, and actions are just processes that happen to those artifacts. So for example, a biological sample artifact is collected, and it might be pooled into a sequencing library and that sort of thing. Majora records how each artifact comes into existence and how all of the artifacts are linked together by processes. And the reason for this is it allows like a full audit trail of where a sample has come from and what's been done with it. And this is what it looks like. This is a screenshot from a testing version of Majora. On the left, you don't need to sort of see this exactly, but it's basically a screenshot that's showing you information about a test sample. It can tell you what the sample's called, sort of information about who it was collected from, what type of sample it is, when it was collected, where it was collected, who told us about it. But the thing that I want to drill down here on the right-hand side in this table, this downstream artifacts table, this tells us that that sample has been incorporated into a sequencing library. That has been, that pooled library has been sequenced, generating some reads, and then a bioinformatics pipeline process has generated a BAM and a FASTA. So we have this sort of end-to-end audit trail of everything that's happened to that particular sample. And the other thing is as well, I'll talk about this a little bit later, but Majora is also the source of truth for quality control. So we are able to programmatically define rules that are applied to the BAMs and the FASTA as they come in. And this can determine whether it's past our basic QC or our high quality QC, or indeed any type of QC that we want to add. And Majora is the sort of source of truth for this. So you can query Majora and say, hey, I want all of the samples that have passed our basic QC, and I want to know where on the login node they are, so I can do some analysis on them and that sort of thing. So many of you probably know what an API is. It's an application programming interface. If you don't know what that is, it sounds really scary, but basically an API allows for a computer program to interface with a human or other computers. You can imagine it as just a means where you send a structured message over the internet and you get a message back. And all interactions with Majora are done like this, and they're done through machine readable APIs. And what do I mean when I say machine readable? They look like this. So these are some, this is JSON. It's sort of a structured, it's a way of structuring text. Basically, these are text files following a basic format that kind of adds structures into text for interchanging information between the API endpoints we use in Majora. And on the left here is an example of sending a biosample to Majora. You can see we've said, oh, this sample was collected in England from Birmingham. It's got some other information about who collected it and when it was collected. And then on the top right, you can see Majora has replied. It says, I've seen your metadata. I've seen your request. That is successful. I've created a new biosample in the database for you. And the interesting thing about that, the important thing about that, is it means that we've actually immediately taken that metadata, accepted it, and it's integrated into the data set. So if you get that message back and it says this biosample has been created, that is there for everyone to query immediately right now. There's no humans getting in the way. So this is a real opportunity for us, because actually this means that we can actually put the validation here. So what if we tweak the collection date for this example on the left and we put it in the future, which is obviously nonsense. You send that request and you get the request at the bottom right here. Majora says there's an error. The success status is false. This sample's garbage and I'm not going to accept it. It's not going to get into the data set. And then it tells you why. It says the collection date has been collected in the future. So now as a user, I can see that and I go, right, okay, yeah, that's nonsense. I should go and fix that. And I know straight away that that sample hasn't been accepted into the data set, but I also know in real time why. So we've done about 15 million of these API requests since we started counting them last May. Last time I checked, which was a couple of weeks ago, we were getting about 72,000 of these API requests every single day for adding biosamples, adding sequencing runs, querying the data set. So we've been operating this back since March. But I said these were machine readable APIs, and obviously a lot of our users aren't machines, they're humans, and they would like a much more graphical user interface to tell them whether their data is good or not. So luckily for me, Anthony Underwood at the Centre for Genomic Pathogen Surveillance built the COG UK sample metadata uploader tool. You can log in through it with Majora, and I'll talk about that later. And you just drag and drop your CSV files into this interface, and it sends them off. So it converts the CSV into the requisite JSON. It talks to Majora, gets the replies back. And then if there's a problem, it'll draw a big, ugly red box around the thing that is wrong. It tells the user, this is really, really wrong. There's a massive exclamation mark. You need to go and fix this. It's not in the data set. And the user can see that, and they'll go and fix it, and they can upload it again. So we've really closed the loop on getting metadata from people and making sure that it's correct. As a flowchart, that's that process that I've described. That's what this is. So on the left, we've got local sites generating FASTAs and BAMs, and they're uploading those to us by SCP or rsync or whatever. And then the metadata is converted through some horrible internal process to CSV. We don't care how people do that. And then they can drop that CSV into the CGPS uploader, and it converts it into JSON. It hits the Majora endpoints over on the right-hand side of this diagram in purple. And then we create the artifacts and processes based on that information. And also for people who are slightly more advanced or want to do some automation, we've got a command line client called Ocarina, and you know that you're using Ocarina because it prints a giant ASCII Ocarina every time you use it. It's got all sorts of command line options that let you set all sorts of things. And you can use it to do all of the standard operations that people want to do with Majora, like uploading samples or querying samples or uploading sequencing metadata and so on and so forth. And we've got some really beautiful Majora documentation I worked really hard on that's completely automatically generated. So it shows all of the correct command line options and examples and all that sort of stuff. And so I mentioned earlier that people can log into the uploader through Majora, and the way we do that is actually through OAuth, which is an industry standard way of doing application authorization. And you might not have heard of it, but you've definitely used it. Anytime you're asked if you want to log into something with, say, your Google account, you've used OAuth. And effectively what we're doing here, Majora is like the identity server. So whereas Google is the identity server when you're using that, Majora is our identity server, and we can direct people to authorize applications inside of COG using Majora. And this means that there's a centralized account system. Anyone can add applications, and OAuth is a very trusted means of doing this as well. So we have a place to store consensus and alignment sequences in the form of CLIMB, and we have a place to store and analyze metadata in the form of Majora. So what we really want to do now is integrate these two sources of data into one coherent data set. And the way that we do that is with a pipeline called ELAN. It's an automated, scalable daily data integration pipeline. It's essentially just an Xflow pipeline. I think it's fair to say if Majora is the brain of COG, then ELAN is probably its beating heart. We run it once every single day to ingest all of the new data. And really, it's more about quality control and checking things are sort of correct and should be ingested into the data set. So it'll check the consensus alignment actually exist, and if they do exist, that they're valid. We'll carry out a whole bunch of QC tests, and we add those metrics into Majora. So Majora is then able to make a decision on whether something's passed QC or not. So we offload that into Majora, so it's not done by the pipeline. And then we link all of the QC pass data once ELAN is finished to a downstream artifacts directory, and that means that the analysts and other pipelines can go and find the data. Cool stuff about ELAN is it operates transparently. So on the left here, I've got an example, a screenshot from Slack. So we have a very well populated COG Slack. So this message is from, feels like a very long time ago now, back in September 2020 when we hit 50,000 sequences, which seemed like an absolutely huge number back then. And basically every day through Majora, ELAN announces, this is the number of samples that I've ingested. This is the number of past quality control. And it'll also highlight there's problems with these files that need to be addressed, or maybe these files are missing metadata, or this metadata is missing these files, and so on. So people get to know every single day whether their data has been integrated into the data set. And then one of my favorite things that we've built for this is that ELAN is now also responsible for triggering other pipelines. So we have an MQTT message broker that people, anything is able to subscribe to and publish messages to. And this on the right-hand side is the first such message that we sent. So ELAN sent a message last July, this was working, and you can see underneath that the filopipe status has been set to started as a result of that message going out. So there's no humans involved in triggering our pipelines anymore. ELAN will just broadcast that it's finished. Anyone who's listening can take advantage of that. So in this example, the filopipe, our downstream phylogenetics pipeline, is subscribed to that ELAN status topic. It sees that it's finished and it raises itself, which is really cool. But it doesn't have to just listen to those messages. We have what I like to call control messages. So one example is maybe a pipeline has fallen over due to some trivial issue, and you just need to raise it and pick it back up again. And instead of having someone log in on SSH and run some commands, or maybe they don't quite know where the pipeline is or whatever, they just send it a message. So you can just send it a message, and I've written a command line client for people to be able to do this. So just send messages to the control channel for a particular pipeline, and you can just say, hey, dust yourself down, get back up, you're needed. And this is actually the first time that someone who wasn't me sent such a message. And then underneath, you can see that the pipeline has responded and said, I've been manually raised by a control message. So it's able to see these messages and take actions based on those messages. And as I just said, so the really important thing about this is that it can be done by somebody who isn't me. So this person who raised this is sort of my protege that I'm training up in the ways of Climb COVID. This was the first time that he managed to get this to raise by himself. So this scary looking flowchart, I'm going to walk you through it. This is sort of really a gist of what happens every single day on Climb COVID. You've already seen the strip at the top. Local sites upload their metadata to Majora over on the right in purple, and they securely copy their sequences to Climb, which is this blue strip on the left. So every single day, ELAN will query Majora through the Ocarina command line client that I told you about. That's in yellow on this diagram. And then it'll try and link those new samples that are to be processed to files that are on Climb that have been uploaded to Climb. ELAN then processes the sequences, so it takes them, it's essentially a whole bunch of SAM tools things chained together just to make sure that the data looks sensible and put it, and if it's not, to try and fix it where possible, put it in the right format, that sort of thing. And then it'll calculate some metrics, and it'll use Ocarina again to update the records in Majora to say, these files, they now exist, we've seen them, we've proved that they're legitimate. I want to add those into Majora, and I also want to add a bunch of metrics to them, and I want you to do QC on them. Once we've done that, we can run some outbound pipelines here in orange. So we can query for, say, high quality data, and we can automatically package it up and distribute it to public databases like GISAID and ENA. And then, of course, there's a whole host of downstream analysis pipelines that I haven't really talked about, but one such one was PhyloPipe, the phylogenetics one. It can generate reports, tables, trees, and it can automatically share them on Climb or to public health agencies here in the UK, or even just publicly via S3. And then finally, we've got a sort of a backdoor, if you will, through Ocarina. You're able to securely access sort of more restricted metadata, and we're able to share that directly with the public health agencies. And the main thing that we do with this, I touched on it at the beginning of my talk. Essentially, we have some cases where samples have a sort of secret name. We're not allowed to know what it is, but obviously it means something to the public health agencies. So we store that linkage to its sort of public COG ID. And we store that on behalf of the public health agencies. And it means they're able to query our data set to work out how to map their internal identifiers to the public COG identifiers. And that's worked out really, really well. So since we began back in March 2020, we've run about 15.5 million compute jobs. And this is just running ELAN, the inbound analysis pipeline. This doesn't even touch on trees or any of the other downstream stuff. And we've used 139 years of CPU time across all of the inbound Nextflow pipelines. And my favorite thing about this chart, obviously, is you can see when we started hitting an exponential growth of the data set back in November last year. It's kind of when we discovered the alpha variant. And this stretches up until March. And what we did in March was we realized this is completely crazy and unsustainable. We need to do some significant hardware and software improvements. And we actually managed to negate the exponential growth of the data, of the daily data set. So we're actually scaling linearly now, even though the data set continues to scale exponentially. So what does 139 years of compute power get you? Well, we've managed to process around 1.2 million SARS-CoV-2 genomes here in the UK since March last year. So on the x-axis of time, stretching all the way back to March. And on the y-axis is the number of genomes that we've processed across time. And you can see that we've sort of hit kind of almost two exponential waves, sort of one from kind of December to March, and things quietened down over the sort of summer period. And now we're heading for an absolutely mammoth winter, I think. So obviously, it goes without saying that this is very serious. These are all real people who've been infected. But I think, it's also just worth saying that, although this is a tragic time, this platform has really empowered us and enabled people to look at genomes at this sort of scale here in the UK, which I think is something to be really proud of. So COG has also been involved in generating a whole host of studies. You can go and look at the COG Consortium website. There's a study looking at how COVID has impacted healthcare. We've obviously worked closely with looking at vaccines and vaccine trials. There's a genomic study which looks at how we can link to the sort of human genome studies. And also, as I was mentioning before my talk, if you were here, I was talking about the ONS, the Office of National Statistics. They do an infection survey, so we work quite closely with them, so they can match some of that data to the genomes as well. Obviously, we have also been responsible for writing a whole host of reports, and where possible, these are completely public and available on the website, and we tend to share these with public health agencies, the UK government, and anyone really willing to listen for the sort of epidemiological insights that we've managed to find from our data set. Obviously, having a data set like this works at small and large scales, so we've been able to do rapid sequencing to detect and confirm outbreaks in all sorts of settings: hospitals, care homes, workplaces, and pretty much all of the academic sites involved in COG have been closely involved with doing this for their own local regions. Certainly here in Birmingham, where I'm based, we've worked very closely with the hospitals to be able to help, answer the question of, hospitals might say, are we having an outbreak right now? We've sequenced these genomes. Can you help us construct a tree and tell us, what needs to be done? And there's plenty of papers. This is one such example from our colleagues in Cambridge. Plenty of papers on how the COG data set has been used to do this. Quite possibly my favorite paper from the COG data set is this one. It's in Science. You might have seen it. It's the dynamics of UK transmission lineage importation. This was basically using the COG data set to answer the question of how did SARS-CoV-2 reach the UK and what happened when it got here? Did it come through the means that we perhaps thought it did? And I think perhaps many people were surprised, not just by how many genomes came through importation from Europe, but also just how many distinct importations there were. This is really something that could only have been answered with the size of the data set that we've enabled people to analyze. There's also a website called MicroReact. This is like a stunning visualization of what's going around in the UK right now. If you looked at it today, you probably don't need to look at it. Hint, it's Delta. But back when things were a little bit more higgledy-piggledy, we could see all sorts of lineages battling for access to our population. And this really showed like a breakdown of where the lineages were moving, how they were moving across time and in space throughout the UK. This piece of software is really interesting as well, because like many things in COG, it's had to go through many different iterations to be able to scale with the size of the data set. Because, when we set out to build this, we weren't imagining 1.2 million samples. Of course, towards the end of last year, I think it probably goes without saying that it was through COG, through our data set, through our close relationship with public health agencies and Lighthouse Labs, and just a real mammoth partnership of everyone in the UK, we were able to identify the first variant of concern, Alpha, towards the end of last year. And things really didn't stop after that. Everything really changed and picked up. And so now I'm on an honorary contract with a couple of my colleagues with Public Health England, and we're providing consulting and help where possible to help them define, well, how can we genomically confirm case numbers? How do we identify variants of concern in the data set? How can we automate that? And so there's many technical briefings now that come out. I've mentioned those at the start of before my talk. Many technical briefings that come out about the data set, and a lot of the stuff in there we've also helped with. And one of the main things that we're working on now is just standardization of these variants of concern. So we're able to put these standard definitions out on GitHub for people to download and use in their software. And essentially, we're just trying to standardize how the variants of concern are called and identified. And the thing about this is anyone can download them, and they know that they're doing it exactly the same way that Public Health England are. So we're trying to standardize this, not just across COG, not just across the UK, but we're sharing this with the world in the hope that it might be useful for other people too. So to summarize the sort of whistle-stop tour of Climb COVID, we've architected and deployed end-to-end infrastructure that supports distributed sequencing at a national scale, but we've managed to do it without imposing too many requirements on the members of the consortium. The infrastructure was built very quickly, but it was built by people with domain expertise. So if we had to do this all over again, we would have made many of the same choices the same way. But I think really the main thing to highlight there is that the prior investment in the Climb computing infrastructure was key in establishing this system. The thing is, Climb already existed. It was already MRC funded. The Medical Research Council in the UK had already funded Climb previously. So the hardware, the staff, the resources, the ecosystem around Climb already existed. And the UK generally as well has quite a good set of science that could be applied to the pandemic through Arctic. Obviously, that's where some of the primers and some of the software for constructing the genomes had come from. So I think the UK was just sort of quite lucky, right place, right time to have Climb, Arctic, and everyone working around it. And I think if Climb hadn't been funded, it would have been almost impossible to build something like this, especially in the time that we did it in. Our use of APIs has enforced a standard interface for providing metadata, and it's also fostered an environment for development of tools against the common platform specification. So this really meant that anyone who built a tool inside of COG could know that it would work on everybody's data because everybody's data looked the same. And I just think that's a really, really powerful, difficult thing to get right, but I think we nailed it. And it just means that, there's a whole ecosystem of tools and people working together on the data set. Our use of messaging queues, as I was talking about earlier, has made it easier to hook pipelines together. So we can have, you don't necessarily need to write glue code. You can just get your program to subscribe to this message queue, and the message queue, we've provided some means for people to have their stuff automatically started based on certain conditions and that sort of thing. And those message queues can sort of communicate pipeline status. So if the pipeline goes down, it'll emit a big message telling everyone, help, I need some help, please raise me again, have a look at my error log. And it also can share telemetry information. So we can capture, well, how long did this pipeline take to run over the past 12 months? And we can go and look at all of the messages. And the main thing as well is it's made it possible for control of pipelines to be relinquished. So, for example, I can spend my time building more pipelines or new pipelines without necessarily having to worry about the ones that already exist, because we've handed the proverbial keys over to somebody who's able to raise a pipeline that's gone down under certain conditions. And they don't necessarily need full understanding or expertise about how that pipeline works, because they just need to know how to craft the right message. The walled garden approach behind Climb COVID allowed fast turnaround and high integrity. So I didn't really touch on this during my talk, but sequences come in, they're ingested into the data set as soon as ELAN has verified them, and that Majora has said that the QC has passed. So you can look at the data pretty much on the same day inside of COG, and then we share it after about a 7-day lag period. And what this does is it means that internally we can use the data straight away, but it also allows us to catch cases where, perhaps some controls haven't worked out the right way, or perhaps the data's been uploaded wrong or the metadata's wrong. And it just means that just before it goes out public, we have that 7-day lag period to make sure that we don't send things up that are quite difficult to take down. So the data that we share publicly is of high integrity. Within 1 week, back in March 2020, we generated more genomes than China, which was the only other real producer of genomes at the time. And within 2 months, we were responsible for half of the world's available SARS-CoV-2 sequences in GISAID, which I just think is a real testament to how quickly the infrastructure that we built was spun up. But not all of the obstacles were technical, and I did touch on this slightly. I think future systems like ours would do well to think about how to generate shareable sample and patient level identifiers. It was sometimes difficult to work out, has this sample come from a patient? Who is it? And I don't mean who by like their name and address, but I just mean, can we tie them to an identifier that means that we can find out what other samples have came from them? And that was a difficult stumbling block, and we're still kind of bumping into that now. But I think either way, this work will definitely lead to, how to build a framework for enabling data sharing between public health agencies and analysts and academia in future. And we did sort of touch on some of that when I was talking about how we have the sort of secure data views for sharing linkage with public health agencies. And I think that sort of, those sorts of ideas will be really, really useful going forward. But for me, kind of my real epiphany of understanding really came from before times when I was a bioinformatician. I felt like I was focused on building the next cool assembler or aligner, or just some really big piece of software that everyone was going to use, and I'd be the next Heng Li and be cited for the next 10 years. But I think what I've really understood is bioinformatics is just so much more than that. It's about just getting everything right, getting the metadata right, getting it in the right format, in the right place. So I'm really proud of what we've managed to achieve through this. And just like from my personal perspective, I've spent about 18 months now working on this, and I've put up about 2,500 code commits to all sorts of non-traditional bioinformatics-y type things, actually writing documentation, writing a lot of glue code, writing API clients, writing obviously Majora and the sort of LIMS systems behind that, and then just loads of other little miscellaneous tools to actually enable people to be able to do the analysis. So that's what Climb COVID really is. It's a platform that's allowed the analysis to happen. But everything that I've talked about sort of been work that I've sort of personally done, but there are loads of other people and loads of other pieces of software that I haven't talked about that are part of COG, an integral part of COG. Matt Bull, Radoslav Pawlowski, and Alex Southgate were really integral in getting Climb COVID off the ground. Rachel Colquhoun, Verity Hill, Ben Jackson, and Anya O'Toole from the Rambo lab were really, really integral in getting the pangolin lineage system together, and also a lot of the phylogenetics pipeline work, which has been extraordinarily difficult. Will Rowe helped build the field bioinformatics toolkit, which is pretty much used by almost everyone to help build the genomes in the first place. And as I mentioned earlier, Anthony Underwood, his team was responsible for building the metadata uploader, which I'm really grateful about because it meant that I didn't have to write any JavaScript for this project. So really with that, I want to say thank you so much for listening. It's been an absolute privilege to talk to so many people about the work that we've built over here. I hope there's some questions, but if you want to read more about what we've done, you might not know, but this was published in Genome Biology a couple of months ago. So we've got a whole paper that kind of details in excruciating detail a lot of the stuff that we've built. I was interviewed on the MicroBinfi podcast, so you can go and listen to me rambling about Majora for half an hour. And yeah, please feel free to check out my Twitter or my GitHub. Thank you very much for listening.
Thank you so much, Sam. That was an excellent, excellent talk. I know that I have a lot of questions, but I figure I will kind of open it up to the audience because I'm sure that everybody else has questions too. So I will let whoever wants to ask questions go for it, and then I'll kind of jump in if nobody has anything. So feel free to unmute yourself or put a question in the chat if you have questions.
Yeah, hello. This is Jamie Pango from the FDA SIP/SAM Bioinformatics Group. Thanks, Sam, for the great talk. Quick question, and maybe I misunderstood it, but it sounds as though, right, you haven't. There's some flexibility in sort of the bioinformatics that people do. It sounded like early in the talk you were mentioning that people make, a consensus sequence where you all haven't sort of locked down, exactly parameters or software that they should use. If that is the case, standardization and harmonization is super difficult, but was there thought given to how, variability in the processing might, result in different results and sort of some inconsistencies there? Hopefully that makes sense.
Yeah, absolutely. Sort of at the beginning, so COG's gone through a bit of a transformation. At the start, it was about 25 different academic sites in partnership with public health agencies, and that's more shifted towards kind of away from the academics and towards larger public health agencies and sequencing labs and that sort of thing. But back when it was sort of more academically driven, there were so many different sites. Eventually, I'd say most of them probably ended up doing the same thing and using the Arctic Nextflow and the whole Arctic pipeline anyway. So there were very few sites not doing that. But again, like you're sort of hinting at, it still needs, quality control still needs to be looked at. And so we actually used to automatically produce quality control reports that would be circulated around everybody in a no blame, no judgment scenario to say, these are what the metrics are for your organization for the sequencing this week. If there's anything wrong, take them down and sequence them again or whatever you need to do. So we did take that quite seriously, but you're absolutely right. Standardizing it is really hard. So really the way that we kind of got around doing it automatically was just reporting it to people and kind of saying, is this right? And if it's not, they can take some action based on that. And I just think that's so much easier than trying to sort of programmatically code in quality control in that way. I hope that answers your question.
No, that's helpful. Thanks again.
Hey Sam, this is Sean. So first of all, fantastic presentation, and I also want to send a big kudos to the whole Cox UK. I mean, here in Minnesota, we started sequencing March 2020, but we wasn't be able to ramp up until Cox UK raised the alarm of the alpha variant. So that was really tremendously helpful, not just from the perspective of Minnesota's sequencing effort, but the whole sort of like United States. But I guess my question for you is, you mentioned about the intake and intake of metadata, sequencing files, and phylogenies are more like a unified national effort, and then each local level or hospital level will be able to sort of like do their own outbreak investigation. And I'm just curious how that process looks like, because you're collecting very minimum metadata, like specimen collection date, country, or zip code. But then for epi investigation, there should be tremendously more PIIs at the very granular level. So I'm just curious how that works.
So
Thanks.
it kind of works in two ways. A lot of the academic sites, especially ours, tended to be near large hospitals and that sort of thing. So literally from my window, I'm like in the shadow of the Queen Elizabeth Hospital here in the UK, which I think is like the largest digital hospital. And so we work quite closely with them in, before COVID, but we worked very, very closely with them during COVID. And what we were able to do for them is take the samples and then trade the results back. And there was sort of two ways this would work. They can either hold on to that metadata themselves. So we can say, well, these are what the samples are. These are what you called them when you gave them to us. And they can kind of do their own internal investigation because they have that rich metadata. But they could also share that metadata with us. And we, if it makes sense, we couldn't then share that with COG. So there's a whole load of different sort of data agreements going on. So although we were uploading samples as a Birmingham site to COG for everyone to analyze, we weren't necessarily sharing all of the data that all of the metadata that we had. We might actually have some of that sort of richer metadata ourselves. And using that, we could help hospitals generate reports and determine whether there are outbreaks or not. But really, it's about closing the loop with the hospitals. And I think that's something that we really want to work towards with COG, because in some cases we had very, very good links with local hospitals and we're able to sort of step in and help them with their analysis, or at least give them the sequences and let their bioinformaticians, if they had any, do it themselves. But in some other cases, there wasn't much of a feedback loop to the local hospitals, and it had to come via the public health agencies, which is just a little slower.
Sure. Thank you. And you're saying that basically, if a local hospital or research institute wants to invest in outbreak, and they want to integrate their PII with the phylogeny, they will be able to somehow download a local build phylogeny of the sequences they just upload, or
Yeah,
they have to reach out to. Yeah, sorry, go ahead.
Sorry. We have software. It's not really from my group. This sort of touches on the work of Andrew Rambo and the postdocs and PhD students in that group. And they kind of do a lot of tooling around phylogenies and that sort of thing. They have a tool called Civet, which you might have heard of, or you might have even used, and it can take a sort of massive tree and take out kind of almost take out a branch, a sort of local branch that might be of interest to you. And it's really that sort of it's kind of like culling things out of the tree that you think might be interesting for a particular outbreak. So, essentially you're saying, I have this sample, we know it came from this ward in this hospital. Does it, if I pull out the branch that it's in, does it contain other people from this hospital, is effectively the question they're looking for. And so it's actually it's quite easy for them to do that if they have a bioinformatician, of course.
Perfect, thank you.
Hi, Sam. This is Kelsey Flork from Wisconsin State Laboratory. I want to say this is a fantastic talk, and I really appreciate that kind of point that you had mentioned earlier about your changing role in bioinformatics and how you see bioinformatics. I've been reading a lot of articles coming out about how just like data science in general has been shifting and changing, and I really see that happening in bioinformatics as well. And I'm sitting here scouring the Climb COVID flow diagram here, and I was just hoping, I could ask so many questions about this, but one of my goals here is to kind of establish a smaller, lightweight version of this here in Wisconsin. And I was just hoping you could speak to a little bit about how you're storing your consensus sequences, your raw sequence data, and how, what kind of database structure you're using to attach those within that Climb structure.
So on the database, Majora is backed by MySQL, so it's nothing particularly special. I think if you're going to go down the route of designing your own software, I would definitely say that the mistake that I made with Majora is I tried to make it really general. And I think I've kind of discovered that there's no such thing as a general piece of software. And so it's ended up with all of these little quirks and hacks in for very specific sort of cases in COG that might have only even happened once. But the problem with the genericism, especially at a database level, is that you end up with a lot of tables representing a kind of inheritance system. I can go more technical if anyone wants as well. But the problem with that is that you get what's called the N+1 query problem, which just means that if you want to fetch, say, a sample, you're hitting the database a whole load of extra times to pull in all of the different pieces of metadata. So that's one thing I kind of take into account. And then in terms of storage, we're using a distributed file system called Ceph, which has proven to be pretty robust. And that's no small undertaking running something like that. And Radoslav is our sort of Climb COVID administrator, and it's just like an absolute machine at making sure these things have worked for the past year and a half. I think what I would have liked to do and what we might move towards is something more like an object store, because actually a file system doesn't really make sense in this scenario. Like, I don't really care about directories. I don't want directories. And it kind of makes it harder for people to get to the data in some ways. So I think an object store would be the way to go. If I had to do something different, I think that's one of the things that I would focus on. I don't know if that answers your question.
No, it does. And actually, object store is kind of the direction I've been leaning towards. So I'm happy to hear that.
I'm going to go ahead and jump in here. Kevin has had his hand up for a while now, so Kevin, go ahead.
No, thank you. Appreciate all the discussions and great talk, Sam. A great presentation, obviously great work. And I don't know if you saw in the chat, but we were fond of your higgledy-piggledy language there, and that's something we'll certainly take to discuss all things SARS-CoV-2 and the challenges in the field that we're all in. But I had a question, I think kind of in line with what James was asking, with the kind of data ingest and like standardization of bioinformatics workflows. So I had a question. I think my understanding outside looking in is that with COG UK, it's predominantly MinION data, and like you mentioned, looking at the field bioinformatics workflow. So I think on the US side, it's actually predominantly Illumina data, and now with a growing presence of MinION data. So in that, I have questions about what is the standard workflow you guys have developed, because I think on our side, we've all kind of been looking at that pending 1.3 release for the field bioinformatics repo, and even on our end, just started making containers to capture the dev branch for the 1.3 to allow for the Medaka models and things like that. And there have been other sort of noted, erroneous indel assemblies that MinION data is susceptible to in that way. So I guess, how are you guys addressing that problem? Are there other resources we should look into? Or is this still the ongoing debate of how to deal with indels with MinION data?
That debate is definitely still ongoing. I've seen the amazing work that you've all been doing with the Docker containers and stuff like that as well. And I think that's almost like sort of light years ahead of the sort of thing that we're doing, because we haven't really, none of this is sort of embracing containers or anything like that. Obviously, we have isolated environments, so like Nextflow lets you run Conda and that kind of thing. So the pipelines themselves are sort of containerized in that way, but it's not quite as powerful as the stuff that you've all been doing, especially in the sort of upstream where the data is coming from. I think a lot of people have just cloned it from GitHub and probably never thought about it since. But yeah, we do actually have quite a lot of Illumina data. Obviously the Sanger Institute is providing mostly Illumina data, and they've probably contributed some 50% of the data set in total. And then I know Public Health Wales, another one of the large contributors, is using Illumina as well. But they're both using a form of the Arctic pipeline that has been written for Illumina data. I don't know a lot more than that, but I know that there's always conversations when new software updates come out and that kind of thing of should we update it, how do we validate it, and all of that sort of thing.
Yeah, and I guess we're like getting to the top of the hour here, but that is a conversation I'm curious to have more the COG UK folks, because with the growing presence of MinION data, we get those issues with the indel events and like database rejections, and kind of curious if you guys have more experience on that, how we can kind of just learn from what you guys have already solved in that way.
Certainly from the database rejection, I don't know if you're like referencing Gisaid there, but we just tell them to stop rejecting our stuff.
I also recently learned that ENA just accepts all assemblies in a different sort of philosophy than the GenBank analog.
And sort of that leads to some trouble, because we submit everything to ENA, who will then go and submit it to the INSDC, i.e. GenBank and Co. on our behalf. And sometimes that doesn't work for some reason, but there's no feedback mechanism there. It's just like you send them this genome and then it goes off into the ether, and you just have to hope that it makes it to the other side out on GenBank. So definitely one of the hardest parts of this has actually been maintaining how to push all of this data out to the public and keep it updated, because ENA is an incredible project, but on some occasions I found where I try and send an API request of like, I want to update this sample, or I want to pull this sample down because it's wrong, and you might get an API response that says, oh, that hasn't been implemented yet, or something's wrong with the XML. I have heard, and I haven't tried it, but I've heard that there's a whole move inside of the ENA to JSON-based endpoints and that kind of thing. So I'm really excited to try those sorts of things out. But yeah, definitely maintaining a sort of a presence in public databases has been really, really difficult. The first couple of first couple of GISAID submissions, I was pretty much doing myself by downloading bits of the COG dataset and then going to the GISAID website and uploading it. And we actually pushed really, really hard for them to give us access to their API so we could do it automatically. And we were probably one of the first people sort of mass testing that. So I was really grateful for that because it meant that I didn't have to do that every Monday.
All right, thanks, Sam.
All right, I know that we are almost at the top of the hour, so I want to be cognizant of people's time if you have to get off the call. But Sam, are you available for maybe a few more minutes for
Yeah,
additional
absolutely.
questions? Okay, awesome, thank you. Yeah, I know that there's probably a lot more. I know that I had a question, and I don't know if it. I guess I'll ask it and see if it makes sense. But so what is your process for updating an existing piece of your workflow? And also what kind of regulations or regulatory bodies do you have to meet and satisfy to ensure your kind of accuracy and quality assurance? I mean, we have like CLIA and everything. So I wonder if you have something kind of a similar regulatory body that oversees all of this.
I think so. On the second question, it's quite a hard one because I think we sort of caught the UK government off guard and built this infrastructure that they probably should have been building themselves. And so really COG has come from an academic project. And in terms of the data that we're getting out, like no one's using this as like this is not a diagnostic test or anything like that. The swabs will get taken up and go to government labs that are hopefully accredited, but as you might have seen in our national news, one of them certainly wasn't. And then once we receive a swab that's been confirmed as positive and it's been extracted, then we do the genomic sequencing. And I think sort of the onus isn't so much on us to have that validated. And some sites will validate their end-to-end workflow, including the bits that we do, but we haven't necessarily been pushed to do anything like that. But I think long term, if we were to continue doing this, there would be talks of things like that. But I think we're quite lucky in the sense that because we're coming after the diagnostic procedure, no one is looking at these genomes like a diagnostic test. And obviously, we're using them to genomically count cases of particular variants, but there's other information that the government is taking into account when we do that as well. We have other types of more validated genotyping tests and that sort of thing. So I rambled for a bit. I'm not sure what your first question was.
Just what is the process for updating any sort of like existing workflows?
So I think this is an interesting one as well because there really aren't that many people working inside Climb COVID. I mean, most of the software that I've talked about is literally just me, and so I'm responsible for checking where the pieces of software that we're using have been updated. And actually, in that chart that I showed you with the sort of exponential growth that we've managed to cut off, one of the things that I did was I just updated Sam Tools. and it turns out they'd reworked, and the reason I updated it was because so I pulled down some data, ran it, tested it, and made sure it looked the same as what it looked like the day before, and that's what gave me the confidence to push the result. But what they've done is completely rewrite how the Sam Tools depth subcommand works and made it about 10 times faster, which was actually an incredible performance boost for the ELAN pipeline. But I think that it's not so much that we follow like a particular set of rules or anything like that. I think everything's kind of ad hoc. When something changes, it's always we're not quite very DevOpsy yet, where we've got a standardized practice of how to handle updates. Updates are more of like a surprise, and we have to treat them very specially.
Thank you. Yeah, that answers my question. So I have a few more questions too, but I want to give other people a chance to ask things. Anybody else have anything? All right, hearing nothing right now. So it seems like you work pretty closely with the Pango team that does the pangolin and the lineage kind of classifications and things like that. I guess what does that involvement look like, and are you just like updating immediately to the latest version that they have pushed in order to do that, and reanalyzing all of the sequences that are in your database?
So they've got so the reason, so the way we keep in touch, I sort of mentioned it at the end. We've got this sort of ragtag group of bioinformaticians inside of COG all trying to work together and keep each other on the same page about things that are happening. It's called the Dippy Group, and we've got like a public GitHub. You can go and read all of our really fascinating minutes and sort of decompositions of bad things that have happened and how they happened. Which I find quite interesting to write. Anyway, that's an aside. And so we kind of use that as a forum where we try and meet at least once a week to talk about things that have changed or things that have gone wrong, and work out next steps. But sort of from a phylogenetics perspective and from the pangolin perspective, I kind of alluded to this in my talk, but sort of the work that I do is certainly more like infrastructure based, and we kind of provide this platform for people to do stuff on, and all of the sort of downstream analysis is kind of independently deployable. It's almost like another team. They can do whatever they see is the best thing to do, which is I think the right way to do things, because obviously they are the best. They wrote pangolin, so they know when the best time to update it is, and I know that they do a whole bunch of internal testing to sort of say, well, it changes some of these lineages, but it makes them better. So we're going to release it, we're going to restamp the data set, and then they'll communicate that kind of to people who are analyzing the data. I think that's worked out really well.
That makes a lot of sense. Yeah, thank you. I guess another question that I had, so you had kind of talked in the beginning that you originally started in kind of wastewater and I guess got microbiome maybe a little bit more. So are you guys going to be kind of looking into wastewater sequencing?
So it's funny you mentioned that, because we do have some wastewater sequencing to be analyzed. So I would like to get my hands on that. Yeah, I think that'd be quite interesting. Something a bit different.
I know that is a program that is really being brought up in a lot of different states here. We have a project that we're working on getting validated, and so it's exciting.
Yeah, it does look like a sort of really interesting way to do surveillance. I'd love to see what we can get from it. So we've done some. There is a study that I'm not part of at the moment that's been doing kind of preliminary analysis. But I would love to get my hands on the data and take a little look at what's in there as well, because that sort of more aligns with my expertise from the before times.
All right. One other question I had, so you said that I think it was Majora is built in Django?
Yeah, that's right.
Okay. Did you look at any other frameworks, or did you just kind of go with that one because you had like expertise in it or something?
Yeah, I'd used it before, so that was my only real driver for picking that. Obviously, it's Python based as well, so Python is sort of in my wheelhouse, and I didn't really want to go and learn like Ruby on Rails or some other whatever sort of du jour framework there is these days. But the thing I like about Django as well is that it's not only been around a long time, but it's got some of the best open source documentation I've seen. So it's always an important factor when I'm looking at something to adopt. So that was really important for me. But yeah, I'm sort of feel like I've fallen behind on it now, because I'm using Django, whatever the latest Django 2 is, because Python 3 was sort of in beta at the time, and they're already releasing a Django 4 sort of alpha release now. So I feel so far behind. So there might be a painful upgrade process to make Majora 3 or 4 to come.
Well, good luck with that. But yeah, no, I totally understand. You work with what you're comfortable with, and
Yeah.
if it works, then yeah, that's
I
great.
think as if anyone else is listening and they're thinking of building something similar, there's nothing really that special about Django. It's just like you were saying then, it's sort of I'm comfortable with it and knew how to use it. But really it just provides you a method for writing models, accessing a database, and kind of wraps some of that annoying stuff away from me as a developer. So if anyone else is wanting to build something similar, but they do like Ruby on Rails or whatever people are using these days, then that by all means that's absolutely the right thing to do. I wouldn't move to Django just because Majora's in it, because there's nothing really special about Django that's made it better for this particular use case.
Good to know. Thank you. All right. Does anybody have any other questions? If not, I think maybe Sam, do you want to? I can maybe send out your email information to everybody on the call after this. And I'm going to go ahead and stop the recording now too.